All right, good morning, good afternoon, everyone. My name is Michelle Tabb, and I'm the Chief Scientific Officer for DiaSorin Molecular. And today I'm going to give you an introduction to our latest product launch, which is our Simplexa SARS-CoV-2 variants direct assay. This assay is our first research use only assay, which is why you see the RUO there in parentheses. And so today I'm going to give you just a flavor for a few things related to this assay. So starting from the next slide, which hopefully my slides will advance. This is always the fun part. Here we go. Okay, so today we'll talk about SARS-CoV-2 variants and also surveillance programs, including some just general information about what is a variant and why we want to test for them. And then I'll give you an introduction to the product itself, the Simplexa SARS-CoV-2 variants direct assay. And then we'll look into melting curve analysis on our liaison MDX. And I'll be able to share with you some of the internal data that we have from testing clinical specimens so far. So starting from the beginning here, what is a variant virus? So we have SARS-CoV-2 here shown, it's 30,000 nucleotides shown here at the top, and all the different genes, many of which you've seen in some of the products that are out there today that make for both the non-structural and structural components of this virus. Then in the end of the day ends up looking like that picture here on the left, including the red spikes, which everyone has seen so many times from pictures in the news. And so just thinking back to even our Simplexa COVID-19 direct product, we target the S gene, the spike gene, which is shown there in red, as well as the ORF1ab, which is shown on the left-hand side in blue. But in this case, we're going to focus in on what's important when it comes to a variant virus, and that is the part of the virus that our immune system sees. And because this is where variants can wreak havoc in terms of our immune response, in terms of the reactions that we have to vaccines and protection from viral infections, okay? So here, in terms of what a variant virus is, we're looking at SARS-CoV-2, we're looking now at the spike gene, which is about 3,800 nucleotides of this 30,000 nucleotide viral genome. And if there's a change in one of the critical nucleotides in that S gene, it leads to a mutation. In this case, if you follow the blue arrow, I'm showing what happens in the case of what you've heard about in the news, the UK variant, the B117 variant, which has a mutation which changes an asparagine amino acid to a tyrosine amino acid. And the way that happens is when there is an A residue in the wild type or normal viral genome that changes to a U. And that actually changes the amino acid that's encoded at that position, 501, in the spike protein. And that leads to a variant in this case. And that can actually have a change in terms of the immune reactivity that this virus would have with our own immune systems. So when a mutation changes a viral genome, it becomes a new variant. But viruses, as we know, constantly change through mutation. We're used to seeing this with viruses like influenza A, for example. So RNA viruses in particular are prone to changes. And a new variant of a virus does occur over time. It's expected to occur through evolution. And sometimes variants emerge and disappear, and other times they emerge and then they persist, which is what we're seeing with some of the variants that are out there today for SARS-CoV-2. So why should we track these variants? Or bother in this case, even looking into them? There's a lot of efforts to sequence them and track them. Well, we need to understand whether the variants can spread more easily from person to person. We need to understand whether or not we're dealing with a variant which can cause milder disease, or especially if it can cause more severe disease than what we're seeing already. And of course, we want to know, is the variant detected by the currently available tests? And for those of you who have been following our Simplexa COVID-19 direct product letters to customers, we have continued to track these variants and look into the primer and probe binding regions for that product to make sure that we continue to be able to detect these variants. We also need to track them because we need to know if there's treatments that are out there, like antibody-based therapies, for example. Will somebody infected with a variant virus still be responsive to those antibody treatments? And then, of course, we need to know the vaccines that have been developed. Will they be effective if we happen to be assaulted with a variant virus causing infection in us? Will we be able to fend it off and not become severely ill? So thinking back to probably earlier this year, what were the first variants that came out? What were the first ones that people were really having concern about? And so you can see there on the left is the original virus that emerged in Wuhan. And then the first variants we started to hear about came from the UK, from South Africa, and Brazil. And those are all considered to be variants of concern. You can see there for the countries that they were first detected. And through next generation sequencing, there's been mutations that are characterized across that whole viral genome, the full 30,000 nucleotides. And you can see there that's there in the next column. And then some hallmark mutations that ended up changing key amino acids in spike protein, which are shown there in the far right column. And what we're looking for in these variants and why there was concern is because, as I just mentioned, more efficient transmission, reduced antibody binding or immune protection, or reduced efficacy in terms of the vaccine. The first thing that we heard about was, whether South Africa virus variant here would be, you'd have protection if you got vaccinated from that South African or Brazilian virus. And so moving a little bit more forward, the CDC went on to further characterize the variants. And so now you hear in the news about variants of interest, variants of concern, and variants of high consequence. And this goes across to the WHO as well. You can find this information also in detail on the CDC website, and they continuously update this website to categorize the variants. And so here, I've shown them in order of increasing severity, with the minimal one being the variant of interest, and then a little bit more concerning would be the variants of concern, and then ultimately variants of high consequence. So a variant of interest is a variant that has a specific genetic marker with changes to receptor binding. That's the receptor to which the virus binds when it's going to enter our cells during infection, or reduced neutralization by antibodies from previous infection or vaccination, reduced efficacy of treatment, potential diagnostic impact, or predicted increase in transmissibility. And so over there on the right-hand side in red, you can see some of the variants there that include New York variant as well as Indian variant and the second generation of Brazilian variant P2. Going a bit more severe is the variant of concern. And so this is where we have evidence of increased transmissibility, evidence of severe disease, reduction of neutralizing antibody treatments, efficacy, reduced effectiveness of the vaccine, or diagnostic detection failures. And there's where we see the Indian variant. We also see the South African variant, the two California variants, B1427 and 429, and the original Brazilian variant. So far, luckily, we have not seen any variants of high consequence, which would be categorized as a failure of diagnostics, a big severe reduction in vaccine effectiveness, or breakthrough cases, or very low vaccine-induced protection. So far, so good there. But of course, this continues to be surveilled for in many different ways, which is what my next slide is about. And that is, what is the CDC doing to track the variants? And just to give you a little background here, there are a lot of sequencing efforts going on and surveillance efforts going on. The CDC is tracking the variants through genomic surveillance, and other health agencies in different countries are as well, including the WHO. But here with the CDC, they're leading what's called the NS3, which is the National SARS-CoV-2 Surveillance System. They're partnering with many commercial diagnostic laboratories just to make sure that they have increased capacity for sequencing and surveilling through next generation sequencing, collaborating with universities as well on the same vein, and also supporting state, territorial, local, and tribal health departments in their efforts to surveil. And they're leading the SPHERES program, which is sequencing as part of the public health emergency response program. So surveillance really is about sequencing. And the best way to sequence this virus, as we've seen, is through next generation sequencing. But that can take a bit of time. It takes usually about 3 days or more in terms of turnaround time. There's only certain capacities associated with the instrumentation too that can run these. And it's extremely costly per sample, on the order of $750 to $1,000 US per specimen. And so in thinking about a more efficient way, or perhaps a more efficient way of looking at surveillance, a solution could be to pre-screen SARS-CoV-2 positive patient specimens to indicate if there's a probability that there's a viral variant of interest. So you could more or less pick or choose specimens that you want to send on for that costly and time-consuming next-generation sequencing. So there are tests out there that allow detection of certain variants, but the additional challenge with many of those is that the currently available RT-PCR variant detection have a requirement for a high complexity workflow, usually requiring specimen nucleic acid extraction and followed by real-time PCR analysis, something that's much more time consuming. And so the solution I want to talk to you about today is the one that we have here at DiaSorin Molecular, and that is for screening with the Simplexa SARS-CoV-2 variants direct assay, where no separate nucleic acid extraction step is required. The assay itself takes about 100 minutes, so a bit longer than our standalone SARS-CoV-2 assay, but allows detection of the mutations associated with some of these variants, which would allow sort of a step in between the detection of positivity and selecting samples for this next generation sequencing or surveillance. So here's a description of our assay. Our assay is RT-PCR coupled with melting analysis, so post-amplification melting analysis. And we use the Liaison MDX instrument as we do for our entire Simplexa product line. The instrument software will generate an amplicon melting temperature analysis and curve visualization to allow identification of the mutations that are listed there. So we have chosen to detect 4 different mutations in the spike gene, and those lead to amino acid changes that are N501Y, E484K, E484Q, and L452R that are associated with potential variants of interest and variants of concern. And so the basic workflow is shown there in the bottom of the slide. So you have our instrument and our direct amplification disk. We provide a test kit with 24 reactions, ready to go reaction mix, as is the format of our other Simplexa kits. And you'll be able to test directly from a nasopharyngeal swab or nasal swab in universal transport media or other transport medias, put into the test, have RT-PCR and melting curves generated, which will then show you if there's the presence or absence of these specific mutations that are associated with variants of interest. So the key here is there is direct detection here without extraction of these mutations in NPS and nasal swabs. And the key also is this is from individuals previously diagnosed with COVID-19. So you're going to want to take a previously positive sample and pull that through this test. This is not to be used, this is research use only. It's not to be used for diagnostic procedures. So it's not to be used as a test that diagnoses patients with SARS-CoV-2. That has to happen with an emergency use authorized test upstream of applying this particular test for variants. So in terms of the intended use, we already just discussed this. So it's RUO, and we detect these 4 mutations, nasal and nasopharyngeal swab specimens from patients with a known SARS-CoV-2 infection. And we couple RT-PCR with melting analysis. The kit there is shown catalog number MOL4350 for the variant assay, and you're also going to want the positive control pack, MOL4360. And I'll talk about that in a moment, but you're going to want to run a positive control every time you run this test kit, and that is for comparison of the melting curves associated with mutations. The product configuration, as you'll see from the instructions for use, is exactly like our other Simplexa product lines. In this case, our Simplexa Variants Direct Reaction Mix has a green cap to distinguish it from the cap color of the standalone Simplexa COVID-19 Direct Kit. And you can see also that in our 4 channels there, we have the different mutation changes, the different mutations that are associated with each of those channels in terms of their detection. So we're able to detect the 501Y in our FAM or 520 channel, the E484K or Q in our JO or 560 channel, the L452R and L-calfluorid channel 610. And then as usual, our internal control will read out in our Q670 channel, the 690 channel. As I already mentioned earlier in the talk, we're targeting the spike gene. We're amplifying the same region that we have actually for our standalone Simplexa COVID-19 product. And we're using our traditional internal control, which is encapsulated RNA template. For the assay definition, you're going to want to run our Studio software version 1.1 or higher. And as I mentioned, also have that positive control pack on hand because you're going to want to put positive control on each run. Our reagent handling and storage is the same as you're used to also for our other Simplexa products in this line for direct amplification. So storing the reagents in a freezer at -10 to -30. And then as you're used to, pulling out a tube of reaction mix, thawing it, and then using it right away to put onto the direct amplification disc together with patient sample. So when a run is set up, as I mentioned, a positive control is mandatory in each run, and I'll talk about why that's important in a moment. But we have the positive control kit shown there at the top, and then we have a kit as we have in our Simplexa other product lines where there's a box of 24 ready-to-use reaction mix files. In this case there, we have a box with brown caps, but you'll see in this product case, there'll be green caps. And so on every run, you're going to want to run at least one wedge that has a positive control and put patient samples there shown with the green dots in the other wedges. I also want to caution you that so far we have not validated direct amplification discs for reuse with this assay. So our suggestion is to wait until you have a batch of samples that you'll want to run on a particular set of these samples, up to 7 at a time, together with that one positive control, filling up all 8 wedges of the disc. Just a couple other special considerations for the assay before I show you some data, and that is that our assay uses high complexity software. So you'll be in the user-defined mode, not the IVD mode. And so this is a high complexity research use only assay, so that does put some more responsibility on your laboratory for using this test. The assay definition can be uploaded by a field application scientist or a scientific affairs staff member, or by using the link that is shown here, and we're happy to provide that for you. You must unzip the file prior to loading into the software on your instrument. I already mentioned that our direct amplification disc here cannot be reused, and so you should consider optimizing the run in a batch style format. And the direct amplification disc in this case does not require the barcode scanning. The scanning will occur when the run is initiated as the disc is spinning within the instrument. I already mentioned the software requirement for 1.1 or higher, and the reason for that in particular is that our software version 1.1 does not allow fluorescence melt curves to be printed on the run report. So if you do want to see those melt curves in a printout, we suggest upgrading to software version 2.0 or higher, which will allow you to see both the melting temperature display and the melting curves themselves, and you'll be able to print those out on the run report. So what you'll see in our instructions for use is a checkerboard chart like this, which shows you the types of variants that can be detected or that are predicted depending upon the results of running the Simplexa SARS-CoV-2 variants direct assay. So on this chart, you can see on the left-hand side, we have the Pango lineage as well as the mutations here that are shown. So just looking down here on the left, we have 501Y, 44K and Q, as well as 452R. And then the Pango lineage of the various variants here, starting from the UK at B117, which has 501Y, and 44K present in some strains, and so on. And I'll just note that these are alignments of these lineages, and sometimes, as you'll see here indicated by asterisks, the mutation has been found in some but not all strains. And so once again, an assay like this can predict that you have a potential variant of interest or concern, but that sample needs to be sent on for sequencing to confirm exactly what type of Pango lineage of variant you have on your hands. And so, and that's just my one note on usage of this product, okay? Okay, so before we look at some data, let's also talk about the assay principle that is working behind this assay. It's a little bit different than the rest of the products, which are qualitative RT-PCR that you're used to seeing in our Simplexa Direct product line. And so here we're using melt curve analysis, which has been a feature that is enabled in our software and has been here this entire time really, and now we're enabling it for the first time with this assay. So first, you'll use the assay sort of through traditional cycling for RT-PCR, which will generate amplicons of the S gene, okay? And that's this process is actually shown in the bottom of the slide here. So say that you have a sample which contains a 501Y mutation, for example. That sample will go into our Simplexa variance detection assay. RT-PCR will occur. And at the end of the reaction, we will do a post-amplification melt analysis going from low to high temperature. So we're monitoring the final reaction through a range of temperatures. And the software will be able to detect changes in fluorescence of amplicons as they melt. And why is this important? Well, during PCR, you're generating a double-stranded amplicon. And in the end of the day, depending upon the particular nucleic acids that are present in that amplicon, it will have distinct features where that double-stranded DNA piece that's generated in the amplicon will melt at a certain temperature, which we call the melting temperature, the TM. So you'll be able to distinguish here is the wild type or normal version of the SARS-CoV-2 virus will have a different melting temperature of that amplicon than when a mutation has occurred. And you'll be able to see that shift in melting temperature. So what's shown on the right is what you would see. So post-amplification, the instrument is able to heat the reaction through a variety of temperatures from low to high. And when that happens, you have fluorescence, which starts very high at lower temperatures. And as the amplicons melt apart, the fluorescence decreases. And that happens at a certain rate and in a certain way, depending upon the nucleotides that amplicon is composed of, whether that's wild type or mutant. And then we do a math trick. Remember back to high school math here, we take our first derivative and we're able to see the exact peak here in blue or red in the bottom of the slide, which indicates the melting temperature of that amplicon that was generated. And through these different peaks, through different melting temperatures, we can predict whether or not there's a mutation present or not. And that's essentially how this melt curve analysis works in the assay principle for the Simplexa SARS-CoV-2 variants assay. So as I mentioned, when you're getting ready to run this assay, you're going to need to load up our assay definition. You can download it from the link that we will provide or have the assistance of a field application scientist member or scientific affairs member. And what you'll see in our instructions for use are the expected temperatures for positive control melting. So once again, those melt peaks I just showed you, we know the ranges that are associated with wild type virus, and you'll also be able to see those with our positive control. So here at the bottom of the slide, you can see for the channel which we detect the N501Y 520, you can see the melting temperature range there is between 60.9 and 64.7 for the positive control, and so on to the other channels for detection of the virus. Okay. So here's our first picture of what this assay looks like when you're running a specimen. This is a clinical specimen containing an N501Y mutation. And so at the top of the slide, just to orient us, we have the positive control, so the wild type virus, so to speak. And if you look at that melting curve analysis at the top, you can see the peaks for each of the channels, 520, 560, and 610. And in this case, we have a melting temperature, which if you compare to the sample profile, which is shown in the bottom picture on the left, you can see that the wild type melting peak is at a certain melting temperature, but this patient sample produced a peak that is farther to the right or at a higher melting temperature than the wild type or positive control peak. And this is exactly what we predict to see when there's the presence of the N501Y mutation. Now, this clinical specimen did not have a mutation show up at the position for E484K or E484Q, which is shown in the channel 560. You can compare it to the positive control there and see that it has a melting temperature about the same as positive control, and so on for the 610 channel, which is where we would detect a change associated with the L452R. And by the way, our internal control is still functioning as you're used to seeing it. It's not based on melting curve analysis or a TM. It's simply based on real-time PCR results and fluorescence analysis for qualitative function of the internal control. So moving on through some other clinical samples here. In this case, actually, we did not have an Indian variant, but we have a G block synthetic piece of DNA here in which we've engineered a double mutant representative of the Indian variant. So this one contains E484Q and L452R. And when we use that particular G block in this assay, we can see in the Joe channel, compared to positive control on the left, we see the E484Q actually have a curve with a TM that's slightly shifted from the pos control, as opposed to E484K, which is the other possible change in that position, which produces a melting temperature even higher, so even further to the right on the curve. So this assay allows you to distinguish in that Joe channel between an E484Q or E484K at that position. There was no mutation present in the FAM channel in this variant, and so I'm not showing that in this picture, but you can also see the presence of the L452R in this particular variant. So we have the pos control for the L452, and then we have L452R with the melting curve that's slightly to the right, a higher melting temperature than the positive control. Moving back to another clinical specimen, we have here a specimen that was sequenced as a Brazilian P1 variant, which is predicted to be a double mutant, N501Y and E484K. And looking at the 520 channel on the left, you can see compared to positive control, we once again see the curve shifted to the right associated with N501Y. And in the 560 channel, the Joe channel, compared to positive control, we see the E484K associated with that clinical specimen. And moving on to the California variant, this was predicted to be B1427 or B1429. And those mutations, California variant, contain a single mutation, which results in L452R. And you can see the only place that we saw the change here was in the 610 channel. I'm not showing you the wild type results from FAM or Joe, but this is the result from that type of a clinical variant. Okay, so that's what the clinical specimens look like. So when you're doing the run analysis and your run is over, you've seen those curves that I just showed you. So what will the run analysis and run reports look like? So following the run, the first thing you're going to look at will be the typical amplification profile or plot report. And what you will see is a bunch of zeros, okay, in each of those channels, which is completely normal, by the way, at least on this plot report. You will not see a CT value in the FAM channel, Joe channel, or CalFluoRed channel for a clinical specimen in this assay, because those channels we're going to wait and look at the melting temperatures in. But what you will see on the report is the results from the qualitative PCR from the typical RNA internal control, which you see here in the Q670 channel. And what you expect to see there is a result. You want to see RNA internal control firing because this monitors for PCR inhibitors, as it usually does in our simplex chemistry. So the report is going to show CT values for all channels, but don't worry when there's a zero in all of those other channels. But you should see a CT value of higher than 20 in the Q670 channel, as you usually do for the RNA internal control. So that's the first validity check that you're going to want to do on the specimens. Next, you're going to look at the melt plot report. And here you'll notice that now the RNA internal controls have all changed to not detected. And now we have actual numbers populating the other channels. It is completely normal for the RNA internal control to say not detected in the melt plot. And the reason is because that is a typical qualitative RT-PCR assay design. It is not meant or intended to generate an amplicon that we're monitoring by melt analysis. Okay. However, we did design the assay to detect TMs and for melt analysis in the other channels. And so for the other channels, you will expect to see a value, which is indicative of the melting temperature. And in terms of references, you'll be able to look back not only at the instructions for use for the typical melt temp ranges associated with a mutation or with a wild type, but you'll also be able to see those curves and compare whether or not you have a shifted melting temperature peak associated with whatever samples you're running compared to positive control. So once again, a TM value will be reported when a melting peak is present. Not detected is going to be what you see in the RNA internal control channel, but I also caution you that in the case that you see not detected in one of the other channels, that's also a type of invalid with this assay, because you're using a patient specimen that was already identified as having SARS-CoV-2 present. And therefore, in each of the channels, you expect to see a result, either for the wild type, the wild type version of the nucleotide at that position, or the mutant at that position during the TM part of this assay. And so I'm going to give you an example to make that a bit more clear. So here, once again, is an interpretation table. You'll be able to see this in our IFU. We can also give you some assistance to look at this or look into this when you're running samples. But as I mentioned, there can be an invalid result, and invalids can come from either the RNA internal control itself, initially in that first amplification not being detected, or invalids can come if you have not detected in the melt analysis and that second window, the results report. In the case of an invalid, you can retest the sample with a new reaction mix file from the same kit, or try to dilute the specimen with UTM, and we're suggesting 1 in 10 dilution, and try to test again. And the reason for this is this assay is a little bit different with a melt temp analysis. And there can be inhibitors present in the specimen that do not inhibit regular qualitative PCR, but may impact our melting curve analysis. And I'll show you an example of that next, okay? So let's look through some clinical specimens, and eventually I'll get to some examples of how to deal with invalids. So in terms of clinical sample evaluation, this is just to show some more internal data that we generated using this assay prior to launch. And so these were 10 clinical specimens that we purchased that actually came from another CE marked kit where there was an N501Y or B117 UK variant predicted. So when we ran those specimens in our test, you can see that our test resulted that there was a mutation present in the N501Y, the 520 channel, but there was wild type readout in the 560 and 610 channels. Our internal controls were detected as they were supposed to be. And so these are valid results. We got results across all 4 of our channels. And then with confidence, we can say that there's the presence of this 501Y mutation. And so these are likely to be UK variants that we'd want to perhaps send for sequencing if we're interested in looking into those. But the assay correctly identified 10 out of 10 clinical specimens associated with the UK variant. Moving on, we have a second set here of clinical specimens, some of which are associated with the South African variant or Brazil variants or another variant. And so in this case, what we see is that in 9 of the 10 specimens, we detected a mutation in 501Y and a mutation in E484K, which is shown there in the 2 hashed green boxes. And so those 9 out of 10 are definitely associated with either South Africa or Brazil variants. However, we also found this one sample, this 2259 sample that's highlighted in the darker blue, where we saw the mutation at 501Y, but it was wild type at the E44K position and mutant in the L452R mutation. And so this is a completely different variant. And so probably is one that's interesting that we'd want to send for sequencing if we saw something like this in our lab. And I'll just highlight that once again, looking back at that reference chart. So if you go across here, if you have N501Y and L452R, actually, we don't see any of that type of variant yet, at least being reported out with a great enough frequency to make the variants of interest or variants of concern list. And so that might be something interesting for your lab to sequence if you happen to detect it with this assay. Moving on, we also have 9 clinical specimens that were associated with the California variant. And so these were obtained right here where I'm sitting in the state of California. But when we ran those samples, we saw 7 of the 9 of them correctly resulted out with detection of the L452R mutation that is associated with the California variants. But in this case, you can see highlighted in pink, 2 of the samples gave invalid results. And they were invalid because their internal control was detected, but we did not detect melting peaks in those channels. So remember, first we need to look at internal control validity. And then the second check is, do I see melting temperature results for each of my 3 channels? And if I don't, I need to consider that sample invalid and probably dilute it to find out what's going on with it to get a result. And so that's exactly what we did with these 2 specimens. We first wanted to know whether or not, well, is the sample just very weak? Is it under the limit of detection of the assay? Or were there inhibitors present in that specimen that needed to be diluted out so we could get a melting temp result from that sample? So we did the investigation. The first check was to take those same 2 clinical specimens and run them in our standalone emergency use authorized Simplexa COVID-19 direct assay. And when we did that, you could see at the top that we got results, perfectly valid results, detected results for the virus. And we had both the S gene and ORF1ab genes detected. So for sure, these samples had SARS-CoV-2, and for sure, they were not under the limit of detection. In fact, these are nice and low and robust CT values. However, when we diluted the specimens 1 to 10, to ask the question here, was there the presence of melting inhibitors? Sure enough, when we diluted the samples 1 to 10, now we see not only that we're detected in an internal control channel for validity, but now we had melting peaks across all 3 channels. And that allowed us to see that the mutation was present in the L452R channel as we expected, associated with these California variants. So this is just a bit of a technical tip for you that if you do see inhibition, at least in the melting temp part of the assay, that our recommendation is to do that 1 to 10 dilution in whatever your transport media is. And so one other thing I wanted to note before I sum up here is that we have already checked the test for its function if you do collect specimens in saline, and the test works very nicely. The melting temperatures are in the ranges that we've already shown in our IFU with saline used as a collection media. We've also checked to make sure that extracted RNA from other testing systems can be used in our assay. My only caution there is make sure that you protect your RNA, perhaps with a component like RNase inhibitor, RNasin, and you can talk to our field application scientists or scientific affairs staff about that if you're interested. Otherwise, just to sum up here, our Simplexa SARS-CoV-2 variants direct assay is a qualitative RT-PCR coupled with melting analysis that allows detection of these mutations at the 501Y 44 position and the L452R position associated with variants and predictive of potential variants of interest or concern, which then you could consider for next generation sequencing. We have this typical all-in-one format where the reaction mix is ready to use. The setup is still in high complexity mode in our software, and this is a research use only assay. So this is a little bit more complex than you're used to with our typical Simplexa Direct products, but still very simple, easy to use, and also takes just a bit over the time of our standalone assay. This assay takes about 100 minutes. The assay definitions are not in a QR code format. You're going to need to download them via that IFU link or with assistance from our field application scientist or scientific affairs. And our positive control must be run when you're going to run patient specimens, as I showed you, because you're going to want to compare the TM values and fluorescent melting curves. Also caution, if you have software 1.1, you won't be able to print those melt curve plots. If you would like to do that, we should consider upgrading you to a newer version of software 2.0 or higher. But as far as we know here, we are going to be the easiest and fastest method for identification of these SARS-CoV-2 mutations that are associated with variants of interest and variants of concern. And this is certainly a very easy to use sample to answer assay that can complement our standalone Simplexa COVID-19 direct assay. And so with that, I'm going to end and I'm happy to take any questions. So Tiffany, I don't know if people can type questions in the box or if they can unmute themselves.
They can actually type questions into the box, or they can ask to be unmuted if they like, and I can unmute them and they can ask the question.
Okay, so I'll give everyone a minute here in case you have any questions that I can answer for you.
There's a question from Becky Fung. Do you still need NTC for the setup?
No, you do not. That is up to you if you want to run NTC, but just like the format of our other Simplexa kits, how you run your controls is up to you to set up individually.
Okay, the next one is, how easy is the distinction between 484Q and K?
So it will be distinct in terms of the melting temperatures, especially with reference to the positive control. So what you will see is that the E484Q will be about 3 degrees higher than the positive control on that run, whereas the 484K will usually be about 6 degrees or higher. So there will be that distinction. Our probe happens to cover this region very, very well. And so we get very nice differentiation of those two. And I also wanted to mention that as far as we know, the E484Q, we may be the only ones out there that identify that and differentiate it in particular, and that is associated with the Indian variant, the B1617 that's circulating.
Okay, and the next one. Are the primers designed to hybridize to target regions imperfectly, i.e. they bind to mutations with more perfect match? And if that doesn't make sense, you can read it in the chat.
So the probes are designed across the region where there is a mutation, but through iteration of design, what we have done is in particular looked to see that the melting temperature, of course, for mutations is higher. And what that would indicate is that the probe is a more perfect match, so to speak, with mutations, or else you wouldn't see that TM shift, because this is a probe-based, like a beacon-based melting temperature analysis. So very good question.
Okay, the next question. Will your results just name the mutation, or will you call out California or UK mutant?
Also a very good question. So this test will only call out the presence of a mutation. And it is up to you as the user then to use that checkerboard type of a box to decide whether or not this is a variant, and to decide whether or not you want to send the sample on for further sequencing to really call out exactly what type of a variant it is. So once again, we're only calling out the presence of the mutation at that position, not of the entire variant status of that particular virus.
Oops. Okay, next question. If the positive control fails, will the entire run need to be repeated?
Yes, because you need to see the positive control curves on that run to compare the TMs to, and to compare your curves to. The positive control is required on every run.
Okay, thank you.
And it shouldn't fail, because it's manufactured by us and it is a virus in universal transport media. So, so call us to help you if that happens.
Next question. How much variation do you see in melt temps on different runs? Is there a need to run positive controls for variants at specific frequency?
Okay, so very, very good question, because instrument to instrument, there can be slight differences in melting temperature. And this is exactly why we want you to run positive control on each run. And by the way, this is not unique to the Liaison MDX system. This is true for every real-time PCR instrument that is out there, right? Every instrument has its own particular qualities. And this is why if you look at our instructions for use, you'll see a melting temperature range associated with each of those channels, even for the positive control. And really, as long as the positive control is on the run for you to compare your clinical specimens with, that should help anchor your results. So you could see those TM differences of the clinical specimens with reference to that positive control. We do not at this point foresee a need to run control variant materials, but that would be up to your lab to establish if you feel more comfortable doing so.
Okay, we have two more questions. Is there a CT threshold above which the variant detection will not work?
Very good question. Okay, so coming into the assay, you'll want a patient sample that is already SARS-CoV-2 positive. If it's coming from our Simplexa COVID-19 direct emergency use authorized assay, we have had success with patient samples that have CT values up to about a CT of 28 or 29, and then pulling them over into this variant assay. And the reason for that is you need a certain amount of virus in order to have beautiful melt temperature detection here. So the idea is that you don't have, this is not like a standalone diagnostic for SARS-CoV-2. So the sensitivity does not go all the way down to 500 copies like it does for a qualitative assay. But the thing here is that in terms of process, this assay is meant to be between identification of a positive specimen and choosing specimens for next generation sequencing. And as has been reported by agencies like CDC, you need this original sample to have a certain amount of virus in it, even to have success in the downstream next generation sequencing, because there's many workflow and processing steps in order to get a high quality sequence. So going all the way back in our Simplexa COVID-19 direct standalone, we have seen success with patient samples that generate CTs up to the high 20s, 27, 28, 29, that will result beautifully in our variance detection assay. We are still working with collaborators right now, and you can also do that in your own lab to find out the success rate of samples going all the way through all 3 steps, all the way through to next generation sequencing. And we can continue to update you on that.
Okay, the next question. Have you seen mixed infections?
So far in the clinical specimens that we have analyzed, we have not, but this is hard to tell because our assay does not call out which variants are present. It only calls out which mutations are present. So if there are actually 2 types of virus in that specimen, we won't be able to say for sure that there's 2 or 3 types or even just 1 type. All that we can say is that at a certain level that there's the presence of the mutation.
And it looks like we have one more, Michelle. Can you discuss the RUO designation and how this might be used in the clinical lab? Any discussions with FDA regarding public health reporting, et cetera?
No, we have not had those discussions. So this is FDA categorized as research use only, not for use in diagnostic procedures. And so as I mentioned earlier, if you want to use this assay to determine whether or not a patient is SARS-CoV-2 positive or not, it is not allowed. That is not a usage of this assay. You should be using an emergency use authorized test, a standalone test for SARS-CoV-2, and there are many of them out there, including our Simplexa COVID-19 direct assay. This assay is meant to be used as a tool for surveillance and also to help perhaps screen for samples that contain these mutations that are associated with variants of interest or variants of concerns that you want to send on for next generation sequencing. So that is all that I can tell you about its status in terms of FDA or public health at this time. Certainly if public health labs would like to try out this assay to see how it can fit into their current workflow and surveillance, we welcome that. It's not prohibited as far as I know, but once again, this assay is research use only, not for use in diagnostic procedures.
All right, and I think that's it for the questions for today.
Okay, fantastic. Thank you everyone for your attention today, and I wish you all a good day and good afternoon. Take care, bye-bye.